Hello, welcome to our channel. I am glad to welcome everyone who joined to watch the video lecture on cryoveterinary medicines. The topic of our lecture today is stem cells cryopreservation. Stem cells are unique cells present in the body that have the potential to differentiate into various cell types or divide identifiably to produce other stem cells. Stem cells are essential cells that replace damaged cells or cells lost due to diseases. These cells are the earliest cells of the cell lineage in all tissue and are found in both embryonic and adult organisms. These cells provide a continuous supply of new cells that make up the tissue and organ of animals and plants. Stem cells have been of great interest as a therapeutic method for various diseases and conditions. The stem cells in embryonic organisms are present in the inner cell mass of the blastocytes, which then differentiated into all other cells in the body. Stem cells in adults, however, are localized to specific areas within the body, like in the bone marrow and gonads. Stem cells are self-renewable. Their potency, capacity to develop into any specialized tissue or organ depend on their origin. The type of stem cells, this is totipotental, pluripotental, multipotental, and unipotent. Totipotent stem cells that can differentiate into embryonic and extraembryonic cell types to construct a complete and viable organisms. These cells are found in the first few divisions of a zygote. Pluripotent embryonic stem cells derive from totipotent cells, which can differentiate into any body cells except placental cells. They are found in blastocytes, early stage embryos that are about 5 days old and consist of 150 cells. Multipotent stem cells, which can only differentiate into a limited number of cell types, usually related to the tissue of origin. These cells are adult stem cells, such as undifferentiated liver cells found in liver tissue, and unipotent cells that can only differentiate into their own cell types but have properties of self-renewal. And depending on the source of the stem cells or where they are present, stem cells are divided into various types. The first is embryonic stem cells, are a group of cells that are present in the inner cell mass of the embryo at a very early stage of development called blastocytes. The blastocytes stage in embryonic development is reached within 4 or 5 days after fertilization, and the number of cells at that point is about 150. These cells are pluripotent, meaning they can develop and differentiate into various cell types during their proliferation. These do not, however, contribute to the extraembryonic cells like the placenta. Embryonic stem cells are present within the embryo, which divided and differentiated into germ layer as they become specialized. These cells have been cultured increasingly as they can be artificially cultured to produce cells of different types. The next type, this is adult stem cells. This is somatic or tissue-specific stem cells. Adult stem cells, also called somatic stem cells, are the cells found in specific tissues that functional to repair and form cells of only the tissues they are found on. These cells are considered less potent than embryonic stem cells as they cannot differentiate to different cell types. Adult stem cells exist in niches or areas created by other cells which secrete fluids and nutrients for the stem cells to remain alive on. These cells are found in certain tissues that undergo continuous cellular turnover. Some tissue, like the liver tissue, however, undergoes minimal division only when the tissue is damaged. Those stem cells are found in both children and adults and mostly localized in tissue like the epidermis, bone marrow, and lining of the intestine. The cells of epidermal layer divide continuously to form new cells as the keratinocytes are shed off. Adult stem cells present in the bone marrow and hematopoietic cells differentiate to form three different types of blood cells and immune cells. Stem cells are also found in the brain that differentiate to form very few nerve cells after birth. The next type, this is induced pluripotent stem cells. The limitations in adult stem cells led to the creation of novel pluripotent cells termed induced pluripotent cells from the adult cells by the process of reprogramming the genes. Induced pluripotent stem cells are formed when the adult cells are cultured with embryonic stem cells, where a fusion of these two types form new cells with stem cell-like properties. Sometimes other somatic cells can also be reprogrammed to acquire pluripotency. Induced pluripotent stem cells are similar to embryonic stem cells in that they can also be stimulated to differentiate into different cell types. However, they are different from embryonic stem cells in the level of gene expression and the conditional of the chromatin of the cells. These cells are of significant importance as they can be used in therapeutic medicine, where a doctor will be able to generate cells of practically all organs of the body for each patient. Besides, they also prevent the use of more embryonic stem cells, which might cause ethical issues. It also helps to study new genetic disease by generation-induced pluripotent stem cells from the adult or somatic cells. The next type, this is perinatal stem cells. Perinatal stem cells are a type of intermediate cells carrying the characteristics of both embryonic stem cells and adult stem cells. They are derived from extraembryonic cells of the fetal membrane, umbilical cord, and amniotic fluid. Perinatal stem cells are known to possess immune privilege characteristic as well as broad multipotent plasticity. Also, these cells simply is isolated from extra embryonic tissue that are typically discarded after birth, effectively avoid ethical issue involvement. where cells are active, not tumorigenic, and are multipotent that can differentiate into cells of the endothelium, hepatic, adipose, and even nerval tissues. The cells obtained from fetal membranes are also not immortal, have a high degree of division to potency. Perinatal stem cells also have research and therapeutic application in the treatment of renal disease, cardiac disease, inflammatory disease, bone regeneration, and the treatment of spinal cord injury. As a result of this application, perinatal stem cells have been cultured artificially to obtain a large number of these cells. The next types, this is mesenchymal stem cells, a type of adult stems or somatic stem cells mostly found in the tissue of muscle, liver, and bone marrow. Human mesenchymal stem cells are the multipotent stem cells with the capacity to differentiate into mesodermal cell lines such as osteocyte, adipocytes, and chondrocyte, as well ectodermal and endodermal cell lines. The most common types of mesenchymal stem cells in the one in the bone marrow, where they differentiate to form the cells of the skeletal system, including bone and cartilage. Mesenchymal stem cells are found not only in fetal tissue, but also in many adult tissue. These are mostly present in small quantities, but are important as they create a niche for the survival of blood stem cells in the bone marrow. Mesenchymal stem cells can be isolated comparatively easily and also produce a higher yield than other stem cells that makes them useful in cell growth, cell differentiation, and restoration of tissue under severe immunological conditions. The potential to produce cells of different cell lines, immunomodulation, and secretion of anti-inflammatory molecules makes these stem cells a useful tool in the treatment of chronic disease. Self-renewal and the capacity to differentiate it into specific cells are the defining properties of stem cells. At the same time, stem cells possess unlimited proliferation potential and pluripotency. However, in steady-state conditions, stem cells behave as slow proliferation cells. In one attempt to define, all the cell population constitute multicellular organisms. Laszlo in 1979 postulated the existence of transit cells that were different from stem cells. These cells are produced by precursor cells population and were short-lived these cells. The time of transit was defined by a maturation process limited their proliferation capacity. He also emphasized that Amplification, which occurs in transit population origination from stem cells, result in stem cells being a minority population, which predict that proliferation rates in these transit cells will be at some point higher than those in stem cells. So, between stem cells and their terminally differentiated progeny are some intermediate cells characterized by a degree of specialization that increase over time, were known as transient amplified cells. The properties of stem cells. Stem cells have the ability to self-renew. Unlike muscle cells, blood cells, or nerve cells, which do not normally replicate, stem cells may replicate many times. When a stem cell divides, the resultant two daughter cells may be 1. Both stem cells, second, two stem cells and more differentiated cells, and three, both more differentiated cells that control the balance between these types of division to maintain stem cells at an appropriate level within a given tissue is not yet well known. Discovering the mechanism behind self-renewal may make it possible to understand how cell fate is regulated during normal embryonic development and postnatally or misregulate as during aging or even in the development of cancer. Such information may also enable scientists to grow stem cells more efficiently in the laboratory. The specific factory and conditionals that allow pluripotent stem cells to remain undifferentiated are of great interest to scientists. It has taken many years of trial and error to learn to derive and maintain pluripotent stem cells in the laboratory without the cells spontaneously differentiating into specific types. The second property, these stem cells have the ability to recreate functional tissue. Pluripotent stem cells are undifferentiated. They do not have any tissue-specific characteristics such as morphology or gene expression patterns that allow them to perform specialized functions. Yet they can give rise to all of the differentiated cells in the body, such as heart, muscle cells, blood cells, and nerve cells. On the other hand, adult stem cells differentiated to yield the specialized cell types of the tissue or organ in which they reside and may have definite morphological features and patterns of gene expression reflective of that tissue. Different types of stem cells have varying degrees of potency. That is the number of different cell types that they can form. While differentiation, the cell usually goes through several stages, becoming more specialized at each step. Scientists are beginning to understand the signal that trigger each step of the differentiation process. A signal for cell differentiation included factors secreted by other cells, physical contact with neighboring cells and certain molecules in the macroenvironment. Stem cells are considered to immortal. They do not age, they can proliferate indefinitely and form any tissue of the organism. One of the mechanisms underlying immortality in the garbage disposal system known as the proteasome, a key node of the proteostasis network. The immortality of stem cell provides the ability for symmetric and asymmetric division. On this slide, you can see schematic duplication of dividing cells. Stem cells divide symmetrically or asymmetrically. Symmetrical stem cells division produce two differentiated cells or two stem cells, which have equal fates. Asymmetric stem cells division produced one differentiated cell and one stem cells which have different fates. So this is the key difference between symmetric and asymmetric stem cell division. Moreover, asymmetric stem cell division predominates during the development phase of an organism. Importantly, uncontrolled symmetric stem cell division can cause cancers. Morphologic things do not always make it possible to draw a clear line between stem and differentiated cells. Therefore, identification and isolation of stem cells rely on specific molecular signature and sulfate antigens marker. Given the high heterogeneity and complexity underlying the stem cell population due to their origin, location, differentiation state, species, the search for new markers is not an easy task, and the list continues to expand. Markers are specific proteins of the surface of the plasma membrane or intercellular structure that are expressed by stem cells only at this stage of development. These markers are called clusters of stem cell differentiations. You can see marker of stem cells. For example, for hematopoietic stem cells marked CD34. For stem and progenitor nerve cells, nestin. A tumor with characteristic marker of embryonic stem cells is OCT4. Adult stem cells, also known as somatic stem cells or tissue stem cells, are a rare population of cells that are found in body through the majority of postnatal life and given rise to limited number of mature cell types that build the tissue in which they reside. Their progeny replace cells that are lost owing to tissue turnover or injury, thus ensuring the maintenance of tissue homeostasis. Well studied example in mammals, including blood, skin, intestine and muscle stem cells, but it's not clear where all organisms contain dedicated tissue-specific stem cells. Currently, the main somatic stem cell used in medicine are hematopoietic and mesenchymal cells. Hematopoietic stem cells give rise to all types of blood and self-renew their own population, found in bone marrow and peripheral blood. These cells are used in the treatment of many malignant or not malignant diseases to replace or rebuild a patient's hematopoietic system, which types of treatment is called a bone marrow or stem cell transplant. Hematopoietic stem cells are also have been used in clinical trials with oversight for the treatment of autoimmune disease, genetic disease, and other indications. This is bone marrow. Bone marrow cells are collected during a surgical procedure that take approximately an hour. The donor or patient often receive anesthesia. Next, doctors used hollow needles to remove the bone marrow from the hip bones. The bone marrow is liquid and looks like blood. Anticoagulants are added to prevent clumping. After the marrow is collected, the cell suspension is passed through a series of sterile filters of decreasing size to remove fat, bone particles, and cellular debris. If there is a delay between collection and infusion, bone marrow may be stored at either room temperature or in refrigeration to 24 to 48 hours. Bone marrow also may be cryopreserved or frozen. If necessary, a procedure commonly used, then it collected for use in an autologous transplant. The frozen cells are usually stored in liquid nitrogen freezing. Peripheral blood progenitor cells. Peripheral blood progenitor cells are another type of cell therapy product that contains hematopoietic stem cells. Cells are collected from peripheral blood using an apheresis device, which acts like a centrifuge to remove whole blood from the donor to separate its component. A needle connected to tubing from the apheresis machine is typically placed in the donor's arm. Blood enters the machine and is processed so that the hematopoietic stem cells and other white blood cells are removed, while the remainder of the blood is returned to the donor through a second needle placed in the other arm. To increase the number of circulation progenitor cells collected prior of apheresis, the donor patient is prepared to mobilize using recombinant hematopoietic growth factor administration. The growth factor is usually administered for several days before cells collection. Patient donation autologic cells also may receive chemotherapy as part of mobilization. If there is a delay between collection and infusion, these cells may be stored at room temperature or in air refrigeration. These cells can be cryopreserved or frozen if necessary. The next type, this is umbilical cord blood. Umbilical cord blood, one regarded as biological waste, is another source of hematopoietic stem cells. Umbilical cord blood can be collected in a safe manner by obstetrical or birthing staff following the delivery of the baby. In other settings, umbilical cord blood is collected into bag. The unit then undergoes further processing, primarily to remove red blood cells and cryopreservation, and is stored in liquid nitrogen freezer for potential use as a later time. Cord blood can be stored in private family bank for potential use by family member or in a public bank to be used by anyone needing a transplant. Cord blood has several advantages as a source of hematopoietic stem cells because the hematopoietic stem cells in cord blood may be less mature. There can sometimes be greater difference in the tissue mass between the donor product and the patient recipient. They also may be decreases incidence of graft versus host disease and decrease third times since units are banked and already types. Patients with rare HLA types are often successful in finding a suitable cord graft than other sources are not available. Cryopreservation of hematopoietic cells. The routine procedure usually involves slow cooling in the presence of a cryoprotectant to avoid intracellular ice formation. The generally accept cryoprotect in dimethyl sulfoxide in concentration 5-10%, which is added dropped at plus 4 degrees Celsius. The cryopreservative solution may also contain serum, animal or human, or seroalbumin. The standard methods of freezing is carried out at a speed of 1-2 degrees Celsius. Heating is carried out at plus 40 degrees Celsius. Dimethyl sulfoxide version is carried out in presence of dextran as an osmotic buffer in a two-step procedure. Mr. Frosty containers are often used for cryopreservation. You can see this container on this slide. Isopropyl alcohol is poured into the middle of such container. Used for freezing hematopoietic cells of the container Mr. Frosty at a temperature minus 80 degrees Celsius, lower temperature refrigeration, provide a cooling rate similar to that used when freezing at a speed of 1-2 degrees Celsius per minute using software freezers. As a rule, the result of such protocol can be compared with the result of freezing with a control rate. Also, current hematopoietic stem cell cryopreservation protocols are clinically effective. Question remains as to whether they are optimal. This is especially important in the storage and therapeutic use of umbilical cord blood for allogenic use when the collect volumes are small and the total number of CD34 cells per kilogram of body weight is crucial for the possible engraftment of the transplant in a recipient. Works related to the optimization of cryopreservation protocol demonstrated that the toxic effect of hematopoietic cells dimethyl sulfoxide in concentration of 5 to 10% at the temperature of its addition plus 4 Celsius degree is minimal. The development of new cryopreservation protocol made it possible to reduce the concentration of dimethyl sulfoxide to 2%, which in turn made it possible to abandon the stage of cryoprotection version with a slight decrease in availability of hematopoietic cells and the degree of their engraftment. The effectiveness of using such cryoprotectant as hydroxyethylated starter and trehalose alone or in combination with dimethyl sulfoxide was also studied. They have shown the effectiveness in cryopreservation of hematopoietic cells, but so far only in laboratory studies. The next type cells cryopreservation. This is mesenchymal stem cells. These are heterogeneous population of neonatal progenitor cells of stromal origin that can be isolated from various body tissues. They are isolated mainly from bone marrow and adipose tissue. Properties of mesenchymal stem cells. Like most stem cells, mesenchymal stem cells are capable of self-renewal and differentiation. On one hand, cell renewal is the ability of cells to continuously divide while maintaining its undifferentiated state. By doing this, the stem cell pool can perpetuate through the organism's life. Mesenchymal cells are considered to be multipotent, meaning under certain conditions, be it in vitro or in vivo, they are able to mature and differentiate into multiply specialized cells. Despite its size, the mesenchymal stem cells play a lot of significant role within an organism. The following are just some of them. The first, this is suppression on immune cells activation. The second, this increase the number of nerve cells. Third, this is reduction of cell disease. Fourth, secretions of neotropic and antigenous factor. Fifth, this is increase synaptic connection, increase the myelination of axons, and increase of number of blood vessels and astrocytes in the brain. Thus, mesenchymal stem cells have both regenerative potential and immunomodulation properties. Clinical application of mesenchymal stem cells are mainly attributed to their important four biological properties: the ability to home to site of inflammation following tissue injury when injected intravenously, to differentiate it into various cell types, to secrete multiply bioactive molecule capable of stimulation recovery of injured cells and inhibition inflammation, and to perform immunomodulation function. Mesenchymal stem cells from bone marrow and cord blood can be cryopreserved using methods used for cryopreservation of hematopoietic cells 1-2 degrees Celsius per minute in the presence of 5-10% dimethyl sulfoxide followed by rapid thawing. Solution for cryopreservation for mesenchymal stem cells usually contain serum, animal or human. An animal carries the virulence of a zoonotic infection. Human is xenogenic. Therefore, researchers are working on replacing these serums, which also reduced the cost of the cryopreservation procedure. For example, with polyethylene glycol and trehalose. With replacement usually allows reduce the concentration of dimethyl sulfoxide to 7 or 2%. But the result of cryopreservation of both mesenchymal stem cells and hematopoietic stem cells in a medium with polyethylene glycol and trehalose were successful only in the presence of 2% bovine serum albumin. The most effective method of cryopreservation of mesenchymal stem cells remains the method in the medium contain 10% dimethyl sulfoxide and 90% serum. The use of trehalose was not effective. Thus, with its potential to develop methods that used xenogen-free cryopreservation solution with low concentration of dimethyl sulfoxide and use a slow cooling rate. While hematopoietic cells and mesenchymal cells are cryopreserved using conventional slow cooling protocol, pluripotent stem cell proved to be significantly more sensitive to slow cooling. Early study using a standard slow cooling protocol, 1 degree Celsius per minute, in the presence of 10% dimethyl sulfoxide, demonstrated modest recovery of human embryonic stem cells, 16-23%, after freezing and thawing, as measured by the number of colonies recovered 2 weeks after thawing. At the same time, the growth rate of such colonies was reduced and the level of differentiations in the colonies was higher compared to control cells. The use of glycerol and ethylene glycol as cryoprotection, both separately and together with dimethyl sulfoxide, showed a low level of colony recovery, 30%. Thus, today human embryonic stem cells are significantly damaged during cryopreservation. The problem of effective cryopreservation of embryonic stem cells is complicated by the cooperative nature of these cells. Unlike adult stem cells, which grow in culture as attaches monolays of unassociated cells, embryonic and induced stem cells usually grow as colonies attaches to a fibrillar layer, inactivated fibroblast. Before cryopreservation, these cells must be separated from the fibrillar layer, transferred into a suspension and cryoprotection added. This procedure results in post-thawing damage. And some words about vitrification of stem cells due to the fact that there are such sensitive cells, embryonic stem cells, egg cells, embryos. Scientists paid attention to vitrification. With slow cooling, ice is formed and the concentration of dissolved substances increasing during the formation of ice, which is the cause of cell damage. Protectants such as dimethyl sulfoxide mitigate this harmful effect by reducing the amount of ice that forms, thereby reducing the harmful increase in solution concentration. During vitrification, cryoprotectants are used in concentrations that completely prevent its formation of ice, 6-8 mole. Vitrification is the solidification of a liquid without crystallization and ice formation. This is achieved either by high concentration of cryoprotectant or by a high cooling rate in which the high viscosity of the solution suppresses the formation of ice nuclei and prevent ice growth. As it cools, the viscosity continues to increase until all molecular motion stops and the solution turns to glass. In this state, the system exhibits the properties of a solid, but retains the molecular structure of a liquid. Vitrification can be achieved by increasing the concentration of cryoprotection to a level sufficient to prevent ice at any cooling rate. However, this requires the administration of much higher concentration of cryoprotectant than who's normally tolerated by cells. The chemical toxicity of cryoprotectant depends on time, temperature, and concentration, and therefore vitrification requires careful development of the composition of cryoprotectant mixture to reduce toxicity and their gradual introduction at increasingly low temperature. However, as temperature decreases, osmotic damage increases, and therefore any vitrification protocol using this approach is often a trade-off between chemical toxicity and osmotic cell damage. There are different methods of vitrification of stem cells. One of these methods was developed by Rubinov et al. They use a cryoprotection mixture of 20% dimethyl sulfoxide and 20% ethylene glycol and 0.5 mole per liter of sucrose. The mixture of cryoprotection is added in stages. Incubation time with cryopreservation is 60-25 seconds at room temperature. To achieve high cooling rate and open elongated straw, a thin elongated capillary with a volume of 20 microliters was used, in which a vitrifying solution with cells is placed. For vitrification, the straw is directly immersed in liquid nitrogen. To thaw, the tip of the loaded straw is immersed in a preheating sucrose culture medium. After thawing, the cells are gradually transferred to solution for washing cryoprotectant containing a decreasing concentration of sucrose. Then they are shown in a culture medium. The use of this method of vitrification made it possible to obtain embryonic stem cells with a variability by the number of colonies of 25% with cryopreservation with slow cooling. The variability is about 5. But vitrification has limitation. This vitrification system requires a lot of manual work, which is the reason for the low productivity of the process. Only suitable for small volumes of cells, such as oocytes, not for stem cells. It requires high concentration of cryoprotection, 6H more, which can have a toxic effect on cells and cause them osmotic shock. Vitrification can lead to the potential infection of cells with pathogenic agents as a result of direct exposure to cell suspension of non-sterile liquid nitrogen with direct contact. Thank you for your attention. My lecture is over. If you have any question, write me, mail on the slide. I will be happy to answer. Goodbye.